start our recording. So hopefully you have found in the Panopto folder the recordings from last week, right? So those are available for you. Don't forget as well, we have those kind of highlight reels available for you as well. So the highlight reels, if you go to the Panopto module, you can see that there's a folder that has already these short little introductory lessons or lectures that are like 15 to 20 minutes long that give you the high points of that particular chapter. So for instance, we're starting cardiovascular system, the cardio part this week. So it probably would have behooved you, would have been good to look at that highlight reel to get just the general layout of what we're going to be seeing coming up this week. So not a problem if you haven't done it, but at least that gives you a snapshot. It gives you a framework of what to expect without jumping both feet into all the detail, which we're going to do anyway. But those can be helpful for people to give kind of a broad perspective. Start simple with the foundation and the framework with things like that, like reading the chapter summary, looking over the lecture notes that are in outline form, not all the detail at the beginning. And that's one of the resources that we have there too. Okay. So, like we were saying last time, we were finishing up with talking about clotting cascades and things like that. We said that there is a way that we form the clot. So we have this positive feedback cycle that ends up activating fibrin. But we also said that there is a separate chemical pathway that's activating a different plasma protein that is called plasminogen. And plasminogen, when activated to become plasmin, remember we take the ogen part off of it when it becomes activated, just like fibrinogen becomes fibrin. When plasminogen gets activated at the same time as the whole clotting cascade, it takes a while to get fully activated, and by the time plasminogen becomes plasmin, that plasmin starts to dissolve the clot, starts to take apart the fibrin threads, dissolve the clot and remove some of those pieces, and hopefully by that time the blood vessel is actually healed, and so it's time to remove that clot, get rid of those components. So we have these things that are floating around in the blood that work to remove clots. We also have things that are floating around in the blood that work to prevent erroneous clots from happening. But they do sometimes happen. So we talked about like deep vein thrombosis and things like that. And so this term that you see here is, when we talk about clots, a thrombus is a clot that develops and persists in an unbroken blood vessel. So this is an errant or mistaken clot that is formed. So like we said, deep vein thrombosis, DVT, is talking about a thrombus, a clot that is formed because maybe the blood's moving too slowly through that bent limb or that bent blood vessel, or there's something that has started the clotting cascade when there has not been an actual break where it should be forming. So these, certainly, if you have a clot inside of an unbroken blood vessel, it's going to restrict the flow, right? It's going to make that tube much more narrow. And so if it occurs in, for instance, a blood vessel in the brain or a blood vessel in the heart, where we have critical tissues that need that blood flow on a regular basis, then it can lead to death, possibly, or necrosis or death of that particular tissue that's normally being fed by that blood vessel. So it can be very critical. Now, if you have a thrombus, that's an erroneous clot that's formed on the side of an unbroken blood vessel. There's a lot of blood flow going by all the time. It's possible that that thrombus can break off, and now it's mobile. It's moving around the bloodstream instead. In that case, we call that an embolus or an embolism. So if you have a thrombus that's floating around, now you have an embolism, and again, that's floating around. It's not a problem when it's in big blood vessels, right? It's just a small piece of a clot floating around in a big blood vessel, like an inferior vena cava or whatever. That's not a problem. But once you get down to a blood vessel that is a smaller diameter, like an arterial or something like that, now that embolism is going to be big enough, it's going to act like a cork in that blood vessel. And again, now everything downstream from that blood vessel that was being supplied previously, now that bloodstream and that blood flow has been cut off. So again, downstream from that, especially if it's in the heart or the brain, you can have death of that tissue very quickly. So embolism is going to be a big problem. Now, it certainly can come from a broken off piece of a clot, but there are other types of emboli. So there are gas emboli, and gas embolus is when, particularly for people who scuba dive, like marine biologists, right, when you talk about getting the bends, what's happening there is when you go down at depth, a lot of the gases that are normally in solution stay in solution. When you come up, if you come up too fast, it's like opening up a carbonated beverage, right? All of that carbon dioxide that's in a can of Coke or something like that, if you look at it, like if you have a glass bottle, you can't really see those bubbles. It's in solution. Now you release the pressure and it fizzes, right? The gas comes out of solution. So when you're diving, those gases are in solution. If you come up too quickly, you get bubbles that will form in the bloodstream. So that's called a gas embolism. The same thing can happen, right? Totally fine if it's going around in big blood vessels, but at some point with that diverse branching pattern that occurs in your circulation, it's going to end up in a blood vessel where it's big enough that it's going to cork it. And if it's something significant, like we said, like brain and heart, then it can lead to significant issues. So you can get gas emboli. You can actually get ones that are, like we talked about, there's fatty plaques that can form in blood vessels. You get one of those that break off, that also can count as an embolus. So it's not only broken off, erroneous clots that have formed, but that's one of the reasons that we can get a freely floating blood clot. So embolus is going to be something that we have to watch out for. So like we said, in terms of undesirable clots, these a thrombus or an embolus, there are different substances that can help with that. Sometimes you hear about these. You hear about taking a baby aspirin each day. So in the clotting cascade, this aspirin can act as what's called an antiprostaglandin, which basically decreases the amount of clot formation. So if you're prone to that as you get older, if you have cardiovascular diseases, they may recommend taking low levels of aspirin to decrease that clotting cascade. Heparin is an anticoagulant. So in that case, if you come in, right, a coagulation is what we're talking about, is clotting. And so if they find that you have had some sort of a clot that is blocking off blood flow, they may add this to you that can help to dissolve the clot. In addition to your normal plasmin, heparin can be used as an anticoagulant as well. And then warfarin, also known as Coumadin, so that is used as well in terms of a medicine. So if you've had somebody who's had an artificial joint or something like that, knee replacement, hip replacement, something like that, oftentimes the clotting cascade, right, you have blood vessels right next to it, there's a chance that, again, that's something that it doesn't recognize. Normally it sees the epithelial lining of the blood vessels, and if it sees anything different, that can set off the clotting cascade. So warfarin or Coumadin, and there are other ones as well, they can use these to prevent, again, it's kind of, they call it a blood thinner, but again, it keeps the clotting cascade from being quite so vigorous. Now, there's pros and cons to that. Number one, you're not going to tend to form clots erroneously, like attached to these different parts that have saved your life, or at least made your life easier. But at the same time, if you do get a cut, you're going to tend to not clot as easily. So it's going to bleed a lot longer than you normally would if your clotting cascade wasn't quite as inhibited. So that leads to this idea and topic of hemophilia, right? So if you have some sort of condition, whether hereditary or not, where you don't have one of those clotting factors in that chemical sequence that leads to the activation of fibrin, then obviously you can bleed out, right? So hemophilia, these are bleeding disorders. So you may lack a particular factor. So you see factor 8, factor 9 here, factor 11. Doesn't matter. You don't have to memorize different types of hemophilia or the factors, etc. But it's a domino effect. If one of those factors along the way is not there or not functioning properly, you're not going to get to the result where you have fibrinogen being activated to form fibrin so you can form clots properly. So this is one of the examples that you look at, like in Bio 181, where you're looking at, the whole family tree, etc., and you're looking at genetic disorders or the heredity. So you can look at some of the patterns of hemophilia, like in some of the royal families in England and Europe, where it's been shown to be passed on through time. But it's just related to, like we talked about, in terms of clotting cascades and maybe problems with it. Okay, so blood transfusions, certainly interesting because they relate to this cardiovascular system where we have, in this case, a reduced volume. And so you may need to infuse it with an additional blood volume to increase blood pressure or an addition of red blood cells. So if you start to lose more than 30% of your blood volume, then you can go into what's called shock. Now, when we talk about shock, it's not like, ooh, I'm so surprised, I'm feeling faint. That is different, right? That's when we're talking about emotional shock or whatever. Cardiovascular shock, we'll talk about it later on when we get to talking about full cardiovascular function. But cardiovascular shock is when there is a reduction in blood flow and blood pressure so that your different organs are not getting the delivery of blood that they need. So there's different ways that you can have this reduction in blood flow, but that's what we're talking about in terms of cardiovascular shock. This is one, it's called hypovolemic shock, low volume shock. You just don't have enough fluid going around in your system. So the heart's doing its job and the blood vessels are doing their job in terms of routing things around, but you're just running a couple pints low. It's like running your car low on oil, right? It's not going to be able to function properly. So blood transfusions are where we're going to add somebody else's blood into your blood. And we've talked about this a bit already when we were doing lab last week. There are, in some cases, whole blood transfusions, but this is in an emergency situation. We said that if you're doing that directly, whole blood from one person to another, then you're getting everything contained within their blood. You're getting their waste products, you're getting their hormones that are floating around, anything else that's in there, you're going to get directly, even bloodborne pathogens or things like that, right? So that's an emergency situation. Mostly, you will get infusions of packed red blood cells. So they've separated the plasma and the white blood cells, and now you have red blood cells only in a saline solution. So when we talk about, for instance, O blood being the universal donor, that's what we're talking about, right? Just red blood cells in a saline solution that is isoosmotic, the same concentration and solutes as your blood, right? Because if we put in whole O blood, we saw you're going to get those anti-A and anti-B antibodies too. You're throwing that into somebody who has A blood, those anti-A antibodies are going to get to work attacking that person's cells, right? So generally, you're getting just the red blood cells that are in a saline solution instead. Okay, so this last topic that we see here is talking about the blood groups, which is what we discussed in lab. Okay, so we're not going to spend a lot of time on this, but you do need to remember that we're talking about these major antigens here, the ABO antigens, where there is an A antigen and a B antigen. The question is, do you have one of those? Do you have the other? Do you have both? Or do you have neither? And then the Rh factor is a separate antigen attached to the surface of the blood cells, and there's only one. So if you have it, your blood is positive. If you don't have it, your blood is negative. Now again, there are more than like 30 varieties of different antigens, but the main ones that determine compatibility are the ABO antigens and the Rh factor. So antigens are attached to the cells. Antibodies are floating around in the plasma. Okay? So you got to remember that the antigens are attached to the cell, the antibodies are floating around. And so you should know, when we look at something like this, you should know for each of the particular blood groups, like we see here, you should know what that means in terms of what antigens they have and what antibodies they may or may not have. Okay? You should also know in terms of lecture, compatibility. Now remember we said that type O cells can be donated into everybody, so we see that here. But if you're talking about, for instance, type B blood, they can get O and they can get B, but they can't get A or AB. Because if they got A or AB cells, their antibodies against type A antigens would start attacking those cells. Okay? So you should know what it means for the 4 different blood types in terms of antigens attached to the cell as well as antibodies. And here, it gives you kind of a distribution, like we said, and how frequent these different blood types occur. So we saw this in our lab results as well. It pretty much reflected, regardless of race or whatever, you pretty much see the same type of pattern in terms of O being the most common, then A, then B, and then you see that AB blood is fairly infrequent. So you have like, maybe we would get 1 out of all the people, all the 24 students, we were getting like 1 AB. We were getting a handful, like maybe 3 B or more, and then A and O were pretty similar, a lot of us, right? Okay, so remember when you were doing the exercise as well, you were looking for clumping. You were adding sera with antibodies in it to see if it would clump like you see here. That's called agglutination. Agglutination is clumping, the reaction of the antibodies with the correct antigen causing that clumping to occur. Okay, so you could have a question on the lab practical asking you, okay, if you saw clumping in this circle and that circle, what blood type is it? Or it could be a question where it says, for type A blood, what does that mean in terms of antigens and antibodies? For lecture, it'd be more like a multiple choice question. You know, type A blood has what types of antigens, and it has them listed out, that type of thing. Okay? RH factor, like we said, is just a question of whether you have that one or not. We said that it was named for the rhesus monkey, where it was originally, ooh, that was a bad square, originally discovered. And so that is our last topic for blood. This table that you see right here is similar to the table in the book. Remember that tables and figures found in the textbook mean that it's a significant enough topic. They're going to write about it, but they're also going to give you some sort of graphic about it, which means it's important. Like this one here, which has the different red blood cells, white blood cells, and platelets, and then what they do. Okay? So that's important information for you as well. Some of the topics that we see towards the end here, I wouldn't worry about in terms of lecture. It's very interesting as you get into nursing or health science fields, etc., how they can rapidly kind of fix a situation where homeostasis is out of whack. But for the most part, we're going to tend to focus on the core information about what are the particular structures, the anatomy, and the physiology. Sometimes we'll relate it to pathophysiology, but we're focusing on what it's like for these structures in a normal body in terms of their structure and function. Okay? Okay, so don't worry about those last couple slides in terms of the lecture. So, do we have questions?